Hello, everyone. Thank you all for being here. My name is Kaylin, and I'm one of the organizers. And for tonight, we have two exciting talks presented by Sophie and Abby. And as Molly introduced, we have an open format, so feel free to raise your hands to ask questions. And for folks online, you can use the Q&A or the chat option to ask questions. And at the end of both of our talks, we'll have our QR code to join our listserv. And we'll also send out resources and link to the YouTube videos afterwards. And so for tonight, it's my pleasure to introduce our first speaker, Sophie Joseph. Sophie was born and raised in Houston, Texas, where she grew up playing tennis, running, and playing soccer. She got her dual bachelor's at the University of North Carolina at Chapel Hill in anthropology and geological sciences, with a concentration in paleobiology. Sophie received her PhD in biological anthropology from Emory University in 2020, where she leveraged ancient and modern genomics to study ancient pathogens, human demographic history, and migration across the world, genetic adaptation to different ancestral environments, and ancient oral microbiomes from fossilized dental plaque. She is now a postdoctoral researcher in the Department of Molecular and Cell Biology at UC Berkeley, with current interests in reconstructing evolutionary events like ancient admixture between humans and other species like Neanderthals. Outside of lab, she still enjoys running, playing tennis, and other activities in the great outdoors, including hiking and paddleboarding. She is an avid traveler and has visited all 6 continents, including Antarctica. So please take it away.
Okay, I'll just project for a second while the microphone's under construction. So thanks so much for the introduction and for everyone coming today, and the speakers, or the organizers, excuse me, for the invitation. I'm excited to talk to you today about some of the insights we can get into our closest human relatives, the Neanderthals, from genetics. So a little bit about my background and where I'm coming from first. As I mentioned, I grew up in Houston, Texas. I'm a lifelong learner and lover of science. I loved going to museums as a kid. I watched the National Geographic Channel, all of those things. But I didn't really have any career academics that were super close to me while I was growing up. And so I wasn't really exposed to the possibility of doing research as a career long term. And so when I went to college, I was essentially an undecided major. I didn't really know what I wanted to do. But through taking some required gen ed classes at UNC Chapel Hill, I fell in love with the study of evolution. And I actually started studying evolution by looking at the fossil record, not looking at genetics. Thanks so much. So as I studied fossils, I went to field school in East Africa in the Rift Valley to study field geology and paleoanthropology. And although I had a great time and it was a wonderful experience, I realized that sort of cataloging bones into plastic bags wasn't really what I wanted to do for the rest of my career, and that is a lot of what it is besides digging things up with a paintbrush. So I wanted to see if I could ask similar questions about evolution, but from a different perspective. And so I decided to try and go to graduate school and sort of make myself into a geneticist. And so that's what I did. I spent my 5 years of my PhD in an ancient DNA lab studying mostly human evolution. And I'm going to tell you a little bit about how that works and what we can learn from it today. So I'm basically a population geneticist. What do population geneticists do? Well, I'm going to have you imagine we have a really big bag or a bucket of these colorful Lego bricks, and they're all different colors. And so population genetics is like looking at our bucket of bricks and seeing, okay, how many red, how many blue, how many yellow, et cetera, bricks are there. But we don't just look at one single brick in the bucket. We actually want to know what is the overall mixture like in our bucket. Are there more blue bricks than green bricks today than there were last year or the year before? where did they go? Why aren't they there anymore? Or where did they come from if they're new? And if I took some of these bricks and I traded them to my friend's Lego bucket, how does that change the mixture at both of our houses of the Legos that we have? And I study a special application of population genetics, specifically using ancient DNA. And ancient DNA is a time capsule of ancient life. So we essentially can get genomes out of skeletal material like fossils and then look at what ancient creatures were like at any point in time that we are lucky enough to be able to get this DNA out of fossils, which is pretty remarkable. So when I say the words ancient DNA, what I mean is just DNA coming from fossilized remains, and then modern DNA is DNA coming from a living person or a frog or some other creature. So modern DNA would be something like this, a dried blood spot card, or it could be a cheek swab or a saliva sample or something else. But ancient DNA we have to get from fossils. Or if we're lucky enough, sometimes we can even use things like mummies as well and look at mummified tissue. But typically, 99% of the time, we are working with bones. So in my lab, I will get bones in the mail from archaeologists, and then I will turn them into powder and extract DNA. And ancient DNA is a little bit quirky because it goes through some stuff when it's in the bones. The fossilization process of bones underground is pretty brutal. DNA is a remarkable molecule, but still over time it degrades and ends up in these short little fragments. And so because of this, we have to do our extraction of the DNA from the fossils in very special laboratories that look like this. This is actually the lab where I did my PhD. So we wear these really weird suits, we wear 2 pairs of gloves, we wear face masks, eye masks, and that's all to actually protect the samples, not to protect us. Because we don't want to be shedding our modern DNA, our skin cells, our hair cells, our sneezes into our precious degraded samples. So here's a picture of me actually drilling into a jawbone of a dog that's from a cave in Bulgaria. It's about 8,000 years old. And so that's what our work looks like in the lab. Everything is done in these little plastic containers to minimize contamination and things like that. So what can we actually learn about ancient DNA from this information? We can look at things like population relationships between past and present individuals. We can try to look at how people or other organisms have adapted to their environment, whether that be their physical environment or their food or pathogens or what have you. We can actually look at how the architecture of the genome has changed and look at mixtures between different species, which is what we'll be talking about today. And we can also look at things like the evolution of pathogens or even the good bacteria that live in dental calculus, because that gets fossilized too. But as promised today, we're going to talk about the Neanderthals, which I think are some of the coolest sort of insights that we've ever been able to get from ancient DNA. So I'll talk through a couple of examples today that I think really highlight how studying archaic ancestry from ancient DNA can be really interesting. The first thing we'll talk about are some genetic insights into their social organization and how their species behavior actually differed from ours. And then we can actually talk about how us having bits of Neanderthal ancestry in our own genomes may have given us some beneficial genetic adaptations. So some of you may know that for most of evolutionary time, we were not alone. So we are over here on the top left. Let's see if my pointer will work. We are Homo sapiens. That is our species. We are all members of the same species. Today, question here? No, oh, sorry. I thought you were raising your hand. I welcome questions if anyone has them. Sorry. No, it's okay. We are the only members of our species left today, but for much of our history, we were not alone. There are many other human-like organisms that existed on the planet throughout time. And perhaps the most famous and well-studied one are the Neanderthals, or Homo neanderthalensis over on the left, but there have been many others. And this whole family tree that I just showed you was essentially constructed entirely from fossil evidence, not from genetic evidence. Besides Neanderthals on that picture I just showed you, we actually don't have any genetic information from any of those other species. So all of this was done based on the morphology or the physical shape of fossils alone. That's in fact how all of dinosaurs have been classified or anything like that. We don't have dinosaur DNA. So paleontologists do the bulk of our work in terms of classifying organisms and how they relate together. But in 2010, the first draft Neanderthal ancient genome was published. This guy here actually won the Nobel Prize in Physiology or Medicine in 2022 for his work on the first draft Neanderthal genome. And we knew for a long time, based on fossils, that we shared territory with Neanderthals, especially in the sort of Europe and Central Asian region. But this sequencing of the draft Neanderthal genome completely revolutionized our understanding of human evolution when it revealed that all of us who have ancestry from anywhere outside of Africa actually have about 1 to 2% Neanderthal ancestry in our genomes. And some of us who are from further east on the Eurasian continent also carry ancestry from another interesting hominid called the Denisovans, who I don't have time to talk to you too much about today, but perhaps I could come back some other time and talk about the Denisovans because they're equally as interesting. But everybody outside of Africa actually has, again, this Neanderthal ancestry from, we think, actually a single mixture event that happened soon after the out of Africa migrations around 60,000 years ago or so. And I'll talk through now exactly how we know that. Yes. Yeah.
I understand the blue area and then the orange area. Can you talk about what's going on in the Americas? The kind of the little colors.
Yes. So the peopling of the Americas began around 30 to 25,000 years ago during the peak of the last glacial max period. And so people were actually able to walk over this area called the Bering Strait. So essentially from eastern Russia into what we call Alaska today. And those people carried ancestry. They were kind of a mixture of East Asian and what we call the ancient North Siberian lineage. And so they actually share this Neanderthal ancestry that all other Eurasians have, as well as some of this orange Denisovan ancestry as well. So they are descendants of kind of East Eurasians, and so thus they actually carry all of this ancestry with them into the Americas. And so that's why the Americas are actually also blue and orange. Great question, yes.
I'm sorry if I missed this. So Africans don't have Neanderthal ancestors of them in place?
Yes, so they actually do have a little bit, like 0.005% or so, and that is actually from backflow from Eurasia into Africa starting around 15,000 years ago, but they did not directly admix with the Neanderthals and Denisovans.
Because they stayed in Africa, you need to go
Exactly.
to Europe, Asia.
Yeah, so the only way to get places was to walk or canoe back in the day, so it was a bit slow going. So the Neanderthals and Denisovans never made it into Africa, and people who stayed in Africa obviously didn't make it out in time to admix with them. We think that Neanderthals and Denisovans probably went extinct by about 40,000 years ago. So the timing between the out of Africa migration at 60,000 and the extinction was relatively short, and so we think that's why that's the case. Great question. Okay. So how do we actually get this Neanderthal ancestry, this 1 to 2% Neanderthal, into our genes? Well, our bodies are all made of trillions of cells. So we have hair cells and skin cells and liver cells and stomach cells. And the remarkable thing about these cells is that every single cell in its nucleus contains the exact same set of genes. No matter what kind of cell it is, our whole genome is actually encoded inside all of our cells. And then the genes get switched on or off depending on what type of cell it is, if it needs to make certain proteins to function or whatever. That's how cells actually become different in their function between skin cells or hair cells or whatever. And our genome within our cells is organized into things called chromosomes. This is the most common human karyotype. So we have 22 chromosomes that most people all have in common, and then most people either have a 2 X's or an XY chromosome as their 23rd chromosome. And we get one of these from each of our parents. This will become important in a second. So that's why there's duplicates of all of them. And then on these chromosomes is our DNA, which contains our genes. Now, let's say we have a blue human, let's say it's a blue mom and a red Neanderthal, a Neanderthal dad, and they decide to have a baby together about 45,000 years ago. After this event, their child will have one chromosome from each parent. We will have a blue chromosome and a red chromosome. Now, humans are really interesting creatures. Like a lot of other animals, we do very interesting stuff when we are doing sexual reproduction. And so when this child makes its sex cells, it will do something called recombination. This introduces variation into the population where part of the chromosome will actually switch places with the other chromosome when these cells are being created. So like sperm and egg cells are being created by the body. So we get part of the red chromosome on the blue and part of the blue on the red. And this happens through many generations. It's been about 1,600 generations since the admixture event, give or take. And so over time, you end up with a genome that's like red, blue, red, blue, red, blue stripes, like really interspersed regions because recombination happens over and over and over. And so we can then pull out these Neanderthal regions, these red regions that are interspersed in modern people, and we can just compare them to our ancient Neanderthal genome and look for some interesting patterns. So let's talk about some of those patterns now. So we know, for example, that Neanderthals probably lived in smaller groups than humans did, and that's because the ancient DNA that we have from Neanderthals has a lot more signals of inbreeding, meaning that they lived in much smaller groups than we did. We also know that females left their natal groups, their birth populations, and moved elsewhere geographically more often than males did. And there's a recent study from a few years ago now that actually shows us an entire Neanderthal family that was buried in a cave together, which I'll talk about now. Okay, so this is the Altai Mountains. Down here on the left is Kazakhstan, down here on the right is Mongolia, and everything to the north, this greenish area, is southern Siberia and Russia today. And this little pin here is Denisova Cave. And Denisova Cave is not only home to some Denisovan fossils, but it's actually also home to a lot of Neanderthal fossils as well. So we know that they lived here at different times. And there's two other close by caves, Chagarskaya and Oladnikov, that are really, really close geographically to Denisova. And so we're going to talk about the Chagarskaya Neanderthals and their family. But first, I need to tell you about a secret other type of DNA that I kept hidden from you until now. So I've been telling you all about nuclear DNA, which is the DNA that we get from both of our parents from each chromosome. But there's other little parts of our cells that do different functions. And the nucleus stores our nuclear DNA. It's in charge of keeping it all wound up. And we have other parts of the cell, like the mitochondria, that are in charge of producing energy for the cells. And there's also some DNA called mitochondrial DNA that exists in these cells. And the funny thing about mitochondrial DNA is that it's actually only inherited from your mother. And the reason for this is very practical. It's because the mitochondria of sperm cells are actually in the tails of the sperm. And so when they fertilize the egg, the tail falls off. And so you're only left with the egg's mitochondria. And so forensic scientists, as well as these archaic ancestry scientists, use this fact to their advantage when they actually found an entire Neanderthal family buried together in Chagarskaya Cave. Now, this is a family tree chart or a pedigree chart. It's sometimes called, you might be familiar with it. The structure of it is that we have two individuals, and then when there's a line descending from them to another individual, that's their child. That's what that means. And so, using genetic information from both the autosomal, the normal DNA, and the mitochondrial DNA, we were actually able to reconstruct a little bit of a family tree for these Neanderthals who lived in this cave. So we know, for example, that between D and H, they have a first-degree relative relationship. So that's either sibling-sibling or parent-offspring or something really, really close. But they carry two different mitochondrial genomes, meaning that they cannot share a maternal lineage. So the conclusion that they made is that Chagarskaya D must be the father of Chagarskaya H, because they're first degree relatives, but they can't be siblings because they would have the same mother, and they can't be mother-son because they would also share the same mitochondrial DNA, et cetera, et cetera. So that's kind of a cool example of how you can actually use genetics to reconstruct even Neanderthal families, not just humans. Another interesting thing about a lot of Neanderthal regions, oh yes, sure, yeah.
Is there any reason that P and H couldn't have the same father but different mothers?
That's also possible, but they would have less, they wouldn't be a full first degree relative. So based on the frequency of genetic variants in the genome, you can actually tell how closely related two people are regardless of their mitochondrial gene. So that's why we knew that they must be like full siblings or full parent offspring. Good question.
From a couple of screens ago, but
Yeah.
you don't need to go back, but
Okay.
for some reason I'm stuck on this. Do we know why the female Neanderthals would there's no theory that
No, so the question was, do we know why female Neanderthals tended to leave their natal groups more than males? This is something that actually varies among human populations as well. So if you look at people who are living today as hunter-gatherers, they will have behavioral or cultural differences between those groups. Will tend to send females out more. Others will tend to send males out more. That's variation within modern humans, obviously. But we don't know why this tended to be the case with Neanderthals. It may have just been a behavioral thing that was sort of shared among people. We haven't found a genetic drive for why this would be the case. Yeah, it's an open question.
Thank you.
Yeah, no problem. Okay, and I'm going to quickly wrap up by just talking about some interesting aspects of the Neanderthal DNA that we actually carry in our own genome. So before I was talking about actual ancient DNA from this family of Neanderthals, but now we're going to talk about how Neanderthal DNA has affected us as modern humans. So our immune systems are really crucial to our survival. And a lot of this depends on our own body's ability to recognize invaders. So that's things like bacteria, that's things like viruses, and also bigger things like parasites. There's a lot of types of invaders. And in humans, there are these things called the toll-like receptors. These are cell surface receptors that live on our own cells, and they are known to detect different types of bacterial, fungal, and parasitic components that live on the surface of those invading cells. So our immune cells are covered in receptors. They look exactly like this beautiful drawing that I made, so I'm sure you can guess. And it turns out that if we look at the density of Neanderthal ancestry across the genome in humans, it's very uneven. And it tends to cluster at high frequency in certain regions, like the toll-like receptors, for example. So what I'm showing you here is a map with a bunch of little pie charts. And these pie charts, when pieces of the pie are yellow, this is populations where they have a lot of Neanderthal ancestry in their toll-like receptor genes. So everybody has these toll-like receptor genes, but some populations are less or more likely to carry Neanderthal ancestry. You'll notice that some of the North Africans also have Neanderthal ancestry in their toll-like receptor genes, and that is that backflow that I was talking about that happened about 15,000 years ago, actually bringing some Eurasian ancestry back into especially North Africans. And so this is kind of makes sense if you think again about how important immunity is to our survival. But in certain contexts, things can actually flip on their head in certain environments, and these Neanderthal regions can also become a disadvantage as well in certain situations. So you may be familiar with this virus. I don't know if you've heard of it. This is the COVID-19 virus, also renamed, I think, aptly as SARS-CoV-2. But it turns out that there's this one genetic variant that really increases the risk of hospitalization for people. And so what I'm showing you here is a bunch of different data sets on the left. So these are all different cohort studies where they looked at COVID-19 health outcomes. was much higher in people who had a certain variant in this very tiny region of chromosome 3. And so it turns out that some people, especially in Central Asia and Europe, have a lot of Neanderthal ancestry at this region. Whereas in other populations like Africans, but also somewhat interestingly East Asians who do have a lot of Neanderthal ancestry but don't carry this variant for some reason. That's an open question that some people are still working on. But there can be a disadvantage as well at having Neanderthal ancestry as your environment changes. So this is a good example of how our species has had to continually adapt over time as our environment has changed in the context of things like a pandemic or dietary shifts or the agricultural revolution. So I think I'll leave it here and go ahead and take questions. You could be first.
I'm sorry, I didn't mean to jump into the question, but just a question about the Neanderthal positive results. I was just wondering if there was any correlation with what happens in human civilization around animal domestication and zoonosis, if that's a correlative factor.
That's a good question. So regarding domestication, there is a lot of Neanderthal ancestry in South Asians and East Asians at the lactase persistence locus, which is the gene variant that allows people to not be lactose intolerant, to digest milk into adulthood. It's most common in Europeans, but it exists at lower frequencies in South Asians as well. And this locus has a pretty high proportion of Neanderthal ancestry in South Asians who do have some degree of lactase persistence in the population, but also it's at high frequency in East Asians who have a very low frequency of lactase persistence in their population. And so it's been theorized that there's probably some secondary immune-related function related to lactase persistence, and it might not just be the ability to drink a lot of milk that increases survival, but there may be some other sort of linked trait going on there as well, given that it's kind of widespread having Neanderthal ancestry at this region, despite lactase persistence not being so common across Eurasia.
And excuse me if I may ask another question.
Yeah, please.
So the Pleistocene was an amazing period of Homo evolution.
Yes.
And it was punctuated by about 30 or so glacial maximum.
Yes.
What is, do we know if that had some involvement in the construction of the hominid brain or anything like that?
That's a good question. So our hominid brain size pretty much reached modern day levels by about 1.5 million years ago, while we still lived in Africa, actually. So this species here, Homo erectus down here, they are by far the most robust and successful member of our genus. They survived for over 1.5 million years on Earth, and they were actually the first hominids to leave Africa. But they had approaching modern human brain size relative to their body size, actually. So they were shorter than us, and they had smaller brains, but proportionally, it was about the same. We also know that we had about the same size brains as Neanderthals did, and we had about the same body size. Neanderthals were actually slightly bigger bodied. So I don't know about glacials specifically influencing brain evolution, but it's likely that they did influence later evolution in Europe, especially the Neanderthal lineage. We also know that the Neanderthals went extinct right before the last glacial maximum, so it's possible that their smaller group size, maybe that played a role in actually the extinction of Neanderthals as well. Yeah, great question. Any other questions? Yeah.
I understand that it's possible to take certain commercially available DNA tests and have your Neanderthal DNA show that. And I was wondering, is that true? And if so, what is the type of test that shows that, or even more specifically, what companies provide such tests?
Yes, let me get a good picture that I think will help illustrate my answer. So the question for those online was about commercially available DNA tests and how they kind of tell you about your proportion of Neanderthal ancestry, for instance, and how reliable they are. So what they do with these commercially available tests, basically it costs about $1,000 to sequence one person's entire genome, so all of their chromosomes to high resolution. So that's what I do in my lab because I am fortunate enough to have grants that support this research. But 23andMe or Ancestry.com doesn't charge you $1,000. They charge you $50 if it's on sale, or $100 or $200 for this test. And so what they're doing is actually sequencing a portion. They like pick random, well, they're not random. They're sort of hyper variable locations along these chromosomes that are known to differ a lot more than the genome-wide average between people. Meaning that they're able to sequence these certain loci, and then what they do is something called chromosome painting, where they try to paint the regions of your chromosomes based on different ancestries that share these like cherry-picked variants in common with you, basically. That's what they're doing. And so they're not capturing the entire chromosome. So when we're talking about the genome-wide proportion of Neanderthal ancestry, if you happen to have a lot of Neanderthal ancestry close to a couple of those locations that they randomly chose, your test may tell you that you're like 10% Neanderthal. I have a couple friends that this has actually happened to before. But if you do bulk actual whole genome sequencing like we do in my lab across Eurasians, it is incredibly consistent, like 1.5 to 2% Neanderthal. So you're kind of getting like a biased sample with these commercial tests. So I would not necessarily believe your Neanderthal proportion always, especially if it's crazy high, because if you actually sequence the entire genome evenly, it doesn't seem like there is very much variation between people.
And this information is only available through the autosomal test versus the Y-DNA and the mitochondria test.
Yeah, typically, yeah, so definitely for mitochondria, and also the Y chromosome is super small, if you notice, so it's very repetitive, the DNA sequences themselves. There's a lot of like CAG, over and over. So it's not like the most informative thing for ancestry. You can get decent information about archaic ancestry from the X chromosome if you sequence the entire thing, just because it's larger. But the Y chromosome, not so much.
Very interesting talk. You mentioned that Neanderthals travel further from the Could you put in context how far they travel and what is the variability of distance?
That's a good question. So we have few Neanderthals that lived close together in terms of where they're buried. So a lot of the sites tend to be cave sites that have actually preserved the remains, and these caves might be hundreds of kilometers apart. And so we've never actually found two like cousins that lived 100 kilometers apart or something. We just don't. There's like been, I don't know, a few dozen sequenced Neanderthals and only 3 at very high coverage that we use very commonly. So we actually don't know. It's not like we could go to all of the Neanderthals and ask what proportion of people are related to each other at that distance. We just don't have that resolution of data. And also, Neanderthals lived for about 400,000 years, and so it's unlikely that we would find like 2 samples of the same age where we could ask the question. So that is very much an open question that we are trying to figure out how to answer, given the resolution of data that we have. Should I defer to? Oh, you have a question? Yeah. Okay.
I have 2 really quick ones.
Yeah.
If a human and a Neanderthal have a baby, is it a human? How? You know what I mean?
Yes, there is actually a hybrid individual that we have. So they are not a human hybrid, they're a Neanderthal and Denisovan hybrid from Denisovan Cave. And so we don't know what to call them. We call him Denny because he's from Denisovan Cave. We just gave him a name as a person. But we actually don't know. In biology, we call this an F1 hybrid, the first generation hybrid. But yeah, so like generally the concept of species as a whole has really kind of been called into question because a lot of people define biological species as non-interbreeding units or populations, and clearly we've been interbreeding regularly with Neanderthals and Denisovans at some point in the past. So it's unclear if we're all just humans at this point. Yeah.
Yeah, thank you. What happened to the DNA in the dinosaur bones?
Mhm.
That you can't do this with them?
That's a good question. So there is some theoretical upper limit for how small your fragments can get before you can't sequence them anymore, which is what we're running up against. Like the average sort of really old Neanderthal DNA is about 50 base pairs in length. And if I were to like extract your DNA in a tube, it would be thousands and thousands of base pair fragments. So we're kind of at the upper limit of our sequencing technology. I think probably for dinosaurs, which are at minimum 66 million years old, they just may be like so broken down that we'd never be able to find them if they're even there anymore. Particularly the chemicals in groundwater, because of their polarity and the polarity of DNA molecules, they tend to like to break or come together and then break apart. Yes.
Among the Pacific Islanders, what percentage of Neanderthal DNA do you have?
Pacific Islanders are very interesting. So they have about the same amount of Neanderthal as like East Asians do, about 2%, but they have up to 5% Denisovan ancestry. Where is my map? Here it is. So over here, you can see down here on the right, they have, it's really variable between 0.1 to 5% Denisovan ancestry. Papuans have the highest proportion of Denisovan ancestry. And then also curiously, there is a population up here in the Philippines called the Aite. They actually have about the same amount of Denisovan ancestry as Papuans do. But we think it's actually explained by mixture with Papuan populations as well. So super interesting population. Actually, my lab mate, Sarah, she's studying these populations, Pacific Islanders, to learn more about their archaic ancestry right now. So it's ongoing. Maybe I'll come back next year and tell you more about it.